And we are back to talk about enzymes, right? Supposedly this is my specialty, so let's see how it goes. Let's go ahead and get started here. So, what are enzymes? Well, I've said this before and I'll say it again. Enzymes are biological catalysts. Whoops. Let's go back. And I forgot to turn on the drawing thing. Let me do that real quick. You have to do all kinds of stuff just to get it so you can draw on your stuff. Here we go. Again, enzymes are biological catalysts, right? Remember what happens with regular catalysts. When we're talking about organic chemistry and hydrocarbon chemistry and all that stuff, where we would use some kind of metal, right, to lower the activation energy required to get over this hump for a reaction to proceed, right? All biological catalysts are the exact same thing. They allow for the lowering of the activation energy, right, so that reaction that would otherwise not be spontaneous, right, like something that you really need to have happen in your body, right, for you to be alive, right, biological catalysts, the enzymes, make it possible for those to happen. So, in this particular reaction I have here, this example, right, this is actually carbon dioxide becoming water, becoming HC3, bicarbonate, and some protons, right? And this reaction is indicative of the enzyme carbonic anhydrase, okay? And carbonic anhydrase makes it easier for you to go back and forth between CO2 and water and HC3O minus and H plus. And it's actually a very important part of your body. This is part of the carbonate bicarbonate buffering system. Carbonate bicarbonate buffering system. In your blood, right? And it's what maintains the correct pH and homeostasis in the blood in your body, right? Without carbonic anhydrase, the enzyme, let's just say you wouldn't be surviving any kind of crazy pH shifts in your blood, right? So that's what carbonic anhydrase does. So remember, what are enzymes? They're biological catalysts, and what they do is that they lower the activation energy, right? So that reactions that otherwise would not be very easy to do become easier to do, so that biological functions can proceed, right? Seamlessly, right? Proceed seamlessly. So that's the important thing there, right? So there are a few different types of enzymes. Actually, let's talk about their naming first. As I was saying before, an enzyme always ends in "ase," right? Literally, the enzyme's name, it will be the name of the substrate that it acts on with the word with the "ase" at the end. For example, sucrase is an enzyme that acts on sucrose, right? Oh yes, so easy, right? So an oxidase is an enzyme that catalyzes a oxidation reaction, right? And of course, there are like many different types of enzymes. Like I was saying before, these are all enzyme classifications, right? And so the oxidoreductases are the enzymes that do oxidation reduction reactions. Transferases, what do? They transfer groups of atoms back and forth between different substrates. Hydrolases, what do? They use water to break bonds, right? Hydrolysis, right? Hydro meaning water, lysis meaning to break something, right? Lyases, right? And this is an enzyme that adds or removes atoms to or from a double bond. Isomerases, that's pretty self-explanatory, right? It rearranges atoms, but specifically it rearranges atoms so that the substrate is still an isomer of the product is still an isomer of the substrate, right? So structurally, the substrate will be different from the product, but after the enzyme works on it, but the formulas will still be the same, isomerase. And then ligases, I'm a big fan of ligases. We use a lot of ligases to splice DNA molecules together. And those use ATP, right, to combine small molecules together. But in my case, I used ligases to combine long molecules of DNA together, right? So those are the classes of enzymes. All right, so where the magic happens, right? Um, I don't know why I put a question mark there. That should be like a where the magic happens, dot dot dot. It's not really a question. This is where the magic happens. The magic happens in this thing called the active site, right, of the enzyme. What exactly is the active site? It's the region on the enzyme, right, that has a shape that fits the reacting molecule, right? And that molecule is called the substrate, the thing I keep on referring to before. What is the substrate? It's the molecule that the enzyme reacts with or acts on, right, to do something. Inside the active site, there are amino acid groups, R groups, that bind to the substrate, right? And some of them actually do chemistry with the substrate. And when the enzyme is completed with the reaction, it releases the products, right? The substrate is now changed into the products, right? So you can see here, we have an enzyme. This is the enzyme part right here, the big bobular, right, thing. And there's the active site. And the substrate's coming into the active site right there, okay? Taking a close look at the active site, yes, this shape kind of does match the shape of the substrate or the molecule that's being acted upon, right? But like I was saying before, serine is actually very important in active sites, right? Here it is doing hydrogen bonding with another serine that's in the substrate, right? There's a hydrophobic pocket right here in the active site, which means that it's a pocket, like a little hole, that has only only nonpolar amino acids like valine, alanine, leucine, and valine again. And since that's there, the substrate has a phenyl ring right there, or benzene, most sticking off of it, that loves to just seat itself right in that little pocket, right? So it sits there, right? And then here we have a lysine, and the lysine is doing an ionic interaction or a salt bridge with the carboxylate that's coming off of the substrate. So I'm not sure what this example is, but it's just an example of a substrate inside of an enzyme. Once the reaction is done, I guess what it does is it makes it so it separates, it pops out, and the reaction is complete. And then the enzyme, since it's a catalyst, it's not actually used up in the reaction, and you can actually use it again, right? You can actually use it again. It can do as many reactions as it wants in the active site once the substrate leaves. So we can describe the enzyme catalyzed reaction as such, right? A substrate attaches to the active site, then the enzyme substrate complex forms, and then the reaction occurs, and then the products are released, and the enzyme is used over and over and over again. So as biochemists, we like to use these little initials here because we're simple, like I said. We have the enzyme, and we have the substrate, right? We add them together, and we get the enzyme substrate complex, and when the reaction is done, we get the enzyme again back into its original form, and we get our product, right? So a good example of this, right, would be an enzyme that actually hydrolyzes the disaccharide lactose, right? It actually breaks that 1,4 glycosidic bond that lactose has, right? Lactose. So the enzyme is named lactase, and so that's the E for the enzyme. And then lactose, right, is the substrate. That's the substrate. The enzyme substrate complex is the lactase-lactose complex. And once the enzyme is complete with the reaction, right, you get lactase back out again as the original enzyme, and then you get two glucoses, a glucose and a galactose, right? Because lactase, the enzyme, breaks Lactase, the enzyme, breaks lactose into its substituent sugars, which is glucose and galactose. Right? So that's that example. There are a couple of different enzyme action models, right? And a very old model is known as the lock and key model. and it sounds exactly like it sounds. It is exactly what it sounds like, right? Where the substrate has a certain shape, right? And then the enzyme's active site has a shape that's the exact opposite of that shape, and it fits in perfectly like a key and lock, right? Forming the enzyme substrate complex, right? Those that A, B, and C there represents different pockets of different amino acids and such, side chains or R groups that will bind to their counterparts over in the substrate, right? But we know now though that that enzymes and proteins and their amino acids are actually very fluid and flexible, right? And so this lock and key model isn't quite. So a more realistic model is known as the induced fit model, right? In the induced fit model, we have the enzyme's active site looking like a different shape than the substrate, right? But as the substrate approaches, it will induce the enzyme through different interactions, the different non-covalent interactions, right? It will induce, using its non-covalent interactions, induce a shape that will fit the substrate. And this particular model is way more accepted, right? So, using lactose as an example again, right? We have the enzyme lactase here, right, with its active sites that are triangular shaped or valley shaped like that. But when the substrate lactose comes in, right, these are hexagonal shaped, and certain interactions, the non-covalent interactions, right, as they get closer, will induce a shape that will fit the substrate perfectly, right? Whereas before it didn't fit perfectly. And when that happens, the enzyme substrate complex carries out the reaction and it separates, right? It hydrolyzes or separates lactose into its substituent saccharides, right? Glucose and galactose, right? So that's what the induced fit model is, right? So that's important. Before we had the lock and key model, where the substrate is opposite the substrate shape is opposite the shape that's inside the enzyme, and it fits perfectly, right? There's no movement there. But in the induced fit model, the substrate will come closer to the enzyme, right? Basically activating it, and once it does that through like different non-covalent interactions that we spoke of before, right? Because they're flexible, then we'll induce a fit of the disaccharide and then proceed with the reaction, right? And again, this is the more accepted form, the more accepted model when it comes to enzyme action. So there are also things called isozymes. One is known as lactate dehydrogenase, right? And what lactate dehydrogenase does is that it actually oxidizes lactate into pyruvate, right? Giving you an NADH, right? Which is just an electron carrier in the metabolic chain, in a metabolic pathway, right? And so there are 5 different isozymes of lactate dehydrogenase, right? There are 5 of them. And each one of them contain a mix of polypeptide units of M and H, right? So the units are M and H. Kind of like when we were talking about hemoglobin before, it was alpha units and beta units. Well, in the lactate dehydrogenase, it's M and H subunits, right? And they're used to convert lactate to pyruvate in different organs, which I already spoke about. In the liver and the muscle, lactate is converted into pyruvate by LDH5, right, which is isozyme 5. The isozyme with 4 M subunits, right? In the heart, the same reaction is catalyzed by LDH2, right? And this one contains 4 H subunits, right? So the difference between LDH1, right, or lactate dehydrogenase 1, and LDH5, lactate dehydrogenase 5, is lactate dehydrogenase 5 has 4 M subunits, whereas its isozyme LDH or lactate dehydrogenase 1 has 4 H subunits. And we'll take a closer look at that in just a second here. So here we can see the different LDHs, right? Here's LDH 1, 2, 3, 4, and 5, right? Sorry, my head's covering that one right there, but this is LDH 5. Right, that's LDH 5 right there. And remember what we said, LDH 5 has 4 M subunits, right? And LDH 1 has 4 H subunits. LDH 4 is in the heart and kidneys. LDH 2 is in the red blood cells, heart, kidneys, and brain. LDH 3, which has 2 H subunits and 2 M subunits, is in the brain, lung, and white blood cells. LDH 4 has 1 H subunit, 3 M subunits in the lung and skeletal muscle, and we already spoke about LDH 5, 4 M subunits that are in the skeletal muscle and the liver, right? We can actually use isozymes like this in diagnostic tools. Here we can actually detect a myocardial infarction, an MI, or heart attack, right? Because of increased levels of creatine kinase and lactate dehydrogenase, right? Creatine kinase is another enzyme. It works with ATP, right? But here we're using lactate dehydrogenase as an example, right? When a myocardial infarction, or an MI, or a heart attack damages the heart muscle, there's an increase in the level of LDH1. You can detect that in blood serum, and that's how you can tell when someone's had a heart attack, right? I had a friend actually, like the other day, right before she was about to go get her COVID vaccine, right? Right before she was about to go get her COVID vaccine, started like having like an anxiety attack about it. And when she got there to the place to get the, I guess here in Arlington, we're doing it at the Globe Life Stadium. No, actually, they've moved it over to AT&T Stadium now, Jerry Jones World. But before, it was at Globe Life Stadium. And when she got there, she was having this what she called was an anxiety attack. But she told us that it was she told us it was an anxiety attack. But then she told us that she was having pain in her arm, and she was having chest pains too, and she thought it may have been heartburn or something from like spaghetti sauce from the night before. And we're like, that doesn't sound right. You should probably go to the hospital and get checked out. And while she was at the hospital, they checked her levels of LDH1 in the blood serum, and they weren't elevated, so she didn't have a heart attack. I thought it was a heart attack, but I'm not a medical doctor. I just know that I've never had a problem with spaghetti sauce causing me pain in my arm. So. But luckily, she didn't have a heart attack, and she got home pretty easily, but with no issues. But that's an example of using LDH as a diagnostic tool, right? Elevated LDH5 elevated LDH5 in the serum, right? That actually indicates liver damage or disease, right? So you can use all these to do a lot of diagnostic tests, right? Just in conclusion, the different isozymes of lactate dehydrogenase indicate damage to different organs in the body, right? Good stuff. So there's some factors that affect enzyme activity, right? And just like we were talking about before, you can use heat and pH and other methods to denature proteins, but proteins are just enzymes, right? They're functional enzymes that carry out reactions. So, or enzymes are just proteins, right? And they're enzymes are proteins, right? They're just functional proteins that carry out reactions, right? So you can use things like temperature and pH, right? Or the presence of inhibitors, which we'll talk about later, right? To inhibit an enzyme's or affect an enzyme's activity. How fast it catalyzes a reaction. So the example I have here is actually of thermophiles that are they can survive in temperatures, bacteria that can survive in temperatures from 50 degrees Celsius all the way to 120 degrees Celsius. That's hot. That's 20 degrees higher than boiling point. So I have a couple of different things to mention here, right? Uh, if you didn't already know, 37 degrees Celsius is actually body temperature, right? And it's actually the optimal temperature that bacteria could grow at. So being able to survive 50 degrees Celsius is pretty impressive. But then, all the way up to 120 degrees Celsius, this is the reason why when we autoclave things, or sterilize them with that big metal machine for like surgical equipment, right? We autoclave things. We autoclave it, and I don't know why I put it two words. It's a single word. Autoclave is two words. It's not two words. It's one word. We autoclave things or sterilize things. At 121 degrees Celsius. Right? Remember I told you that earlier? We do that because extremophile bacteria can survive up to 120 degrees Celsius, and when you autoclave something, you're completely sterilizing it, right? We're talking like killing 99.99999, like I think it's like 69%, right? Is what the CDC says is called sterilization. And to achieve that, we have to do sterilization or treat whatever we're sterilizing 1 degree higher than the life range, temperature range of thermophile bacteria, which is 120 degrees. So that's the first thing I wanted to talk about when it comes to thermophile bacteria. The second thing about thermophile bacteria is that they actually are very valuable to us when it comes to biological research. They're the basis for what we call PCR. Does anybody know what PCR is? Of course you do. It's called the polymerase chain reaction. So a long time ago, I think it was like 1990. Wow, it's funny saying that 1990 was a long time ago, but that was like 30 years ago. This dude named Kerry Mullis, right? And I think he was high on acid or something like that. But he figured out the fact that we could replicate DNA replication. We could simulate. No, not simulate. We could do DNA replication on demand, right? Of target sequences of DNA by using the enzyme that's in these thermophiles, right? That does DNA replication. And the reason why we could do that is because we can control the enzyme by using heat, right? By using heat. So if you were trying to replicate DNA in a human using what we call polymerases are the enzymes. Polymerases are the enzymes that are in living organisms, right? That are living at normal temperature. These polymerases, right, are responsible for doing DNA replication, DNA translation, DNA transcription, all that stuff, right? But since they exist inside us at 98.6 degrees Fahrenheit or 37 degrees Celsius, right? They would die if we try to heat up their temperature any higher than that, and they become inactive. And so we can't be like, hey, human polymerase, go and replicate this DNA for me real quick. Start here and stop here, right? How do we do that? Well, we need to find, Kerry Mullis figured this out, we need to find a polymerase that can be activated and inactivated using heat, right? Or temperature is what I mean. Temperature. And there are thermophiles or extreme thermophile bacteria. One specifically is called Thermus aquaticus. Aquaticus. And it has a polymerase that's heat stable, right? It's heat stable because it's living in freaking, these heat vents here where the temperature is like 120 degrees. Right? So what we can actually do then is in PCR, we can have a strand of DNA, right? And we could target a section of that DNA, like a real important gene, and amplify that part, right? Or replicate that part of the gene, right? By taking the DNA genome, or the coding section of the DNA, out of the cell and putting it into a tube, along with this Thermus aquaticus polymerase, right? And some primers, and using heat to control the uh, the reaction, right? So what are the steps? It's actually really simple. PCR is, right? What you do is, and you've heard a lot about PCR too, because they used PCR was like the premier COVID-19 diagnostic tool, right? That and the antigen test, but the PCR test was far more reliable, right? Because in theory, you only need one, literally, not literally, but in theory, you only need one strand of DNA for it to work, right? And what happens is that the first step of DNA is that you need to denature it. Why do you need to denature it? Well, you need to denature it in replication because you need the enzymes to spread apart. Sorry, not the enzymes, but the double helix to spread apart. The only way to do that, right, in the body is by using special enzymes, right, like helicase, topoisomerase. And what those do is they unzip the DNA so that the bases, the nitrogenous bases, right, the A's, the G's, the T's, and the C's, adenine, thymine, cytosine, and guanine, are exposed, right, so that a different enzyme can come in and make copies. Well, it's very hard for us humans in the lab to control helicase and topoisomerase, right, to denature the DNA enough so that we can get access to the DNA sequence. Well, what do you do then? Well, those DNA sequences are being held together by hydrogen bonds, right? The double helix is being held together by hydrogen bonds. And if they're being held together by hydrogen bonds, what can disrupt those hydrogen bonds? We've talked about this many times already. It's heat. In fact, hydrogen bonds in water are broken at 100 degrees Celsius, right? Well, you can actually break the hydrogen bonds that are holding together a double helix of DNA, right, by heating the temperature up to 95 degrees Celsius, and it unzips the DNA. If we did that with our normal polymerases that are in our bodies, it would destroy those polymerases. But since we're using the polymerases that are in Thermus aquaticus, it can survive that. So after we unzip it, right, with the heat, we can bring the temperature back down to super low. And when it's low, we can add in little molecules called primers that tell the polymerase where to start replicating. And then we can bring the temperature back up a little bit to 72 degrees Celsius. And that is actually the optimal temperature for Thermus aquaticus' polymerase to do DNA replication. This is huge, huge breakthrough. I mean, Kary Mullis, my God, like he came up with this while he was high on something, acid or something like that. I mean, can you imagine? PCR is like the basis, is the basis and the foundation for the last 33 decades of biological, biochemistry, molecular biology research. Right? It's quite amazing. We'll talk a lot more in detail about PCR later on in life. Not later on in life, like next lecture, because we're going to be talking about nucleic acids next lecture. But just know that thermophile bacteria, thermal and enzyme activity can be affected using temperature, and this is one of the examples using the polymerase from Thermus aquaticus, right, to do polymerase chain reaction. Okay. Next, moving on. Oh, temperature and pH effects, right? Well, if you are an enzyme, right, that's inside humans, like I said before, your optimal temperature for activity is 37 degrees, right? So here in this chart, you can see here at 37 degrees, the reaction rate is at its highest, right? If you're too low, it's low. If you're too high, it goes down too. So you want to be at 37 degrees Celsius, right, for the highest activity. You start losing activity at a temperature above 50 degrees Celsius. Hell, if you bring it up to 95 though, like in polymerase chain reaction, it's dead. The enzyme's dead. Other things that also affect the pH, affect enzyme activity is pH, right? So a good example of this is, okay, so we have an optimal pH of 7.4 actually in cells, most cells, right? And so enzyme activity goes up all the way to its peak at 7.4, and then it goes back down when it gets too basic. So a really good example of this is my enzyme I studied back in grad school that I told you about in the last lecture, luciferase, right? The pH is actually what activates and deactivates that lid that makes luciferase allow the substrate luciferin to come in to make blue light. So this stuff is real, right? This stuff is real. So here's some pH values, right, for some optimum Optimal activity, right? 7.4 is what I said a second ago, which is which is the body's optimal pH. We have pepsin in the stomach, right? It breaks down peptide bonds, and its optimal pH is 1.5 to 2.0, which makes sense because it's in the stomach, right? The stomach is actually very acidic, right? That's why when you throw up after a night of drinking, it burns your throat because there's acid in your stomach, hydrochloric acid, I believe, right? Lactase, we talked about lactase already before. It's in the GI tract. It breaks down lactose into galactose and glucose. It's also slightly acidic because just past your stomach into the GI tract, it starts to become more basic, but it's still slightly acidic. Sucrase, the enzyme in your small intestines that breaks down your sucrose into fructose and glucose, also slightly basic. Amylase, it's in the pancreas. It breaks down amylose, right? And it's getting close to neutral pH. Urease in the liver, its substrate is urea, close to physiological pH. Now we're getting a little bit more basic. Remember I said as you get more and more in your small intestine, it becomes more basic. So trypsin, which breaks peptide bonds, which is basically breaking down the amino acids from the proteins you eat, right? Like your steak. Your steak goes in as amino acids that are in the shape of a protein, right? The muscles that you eat from the steak, right? Like myoglobin and such, and actin and stuff like that, right? And myosin, and you eat it, and it goes into, you chew it up, right? Which is the mechanical breaking up of those proteins, but it gets further broken down into its substituent amino acids when it gets into your small intestines, which are slightly more basic than your stomach, right? Where trypsin goes and breaks those down into their substituent amino acids. The pancreas has lipases, right, which break down the amino acids. The lipids. Remember what I said about lipids in our previous lecture. Lipids are base catalyzed reactions, right, to break them down. And then arginase breaks down arginine in the liver, and that's super basic at 9.7, right? So keep all those in mind. So the last thing we need to talk about is enzyme inhibition. There's a few different types of enzyme inhibition, but what it actually is what enzyme inhibition actually is when normally you have the enzyme plus a substrate and you make the enzyme substrate complex, and then after that, like we said before, you get the enzyme back out again as it was before you started, so you can do it again, do the reaction again, and then you get a product, right? That's different from the substrate. You can actually inhibit an enzyme by adding an inhibitor, right? And if you inhibit that enzyme, you don't get a product at all. Okay, you don't get a product at all. This is actually the basis for a lot of drug molecules, right? Back when I was in grad school, I was also developing drugs to combat cyanide poisoning and nerve agents. And one of the things that we were doing was we were synthesizing molecules that were very similar to the nerve agent itself, right? Hoping that they would be able to block the nerve agents or the cyanide, right, from attacking its targets, right? It's a lot more complicated than that, but that's what we're doing. The goal was to create inhibitors for enzymes that were acting that would make you sick from acting with different nerve agents and cyanide. So there are three types of normal inhibition here, right? There is competitive inhibition, and in competitive inhibition, it sounds exactly like it. It is exactly like what it sounds, where a competitive inhibitor goes into the active site and blocks the substrate from going in there, right? It blocks the substrate from going in there. Okay. Uncompetitive inhibition, right? What it does is that after a substrate has already gone into the active site and is going through the enzyme reaction catalysis process and has formed the enzyme substrate complex, an uncompetitive inhibitor will go in and bind to the entire enzyme substrate complex, rendering it useless, right? You don't get a product out of it anymore. And in non-competitive inhibition, the substrate tries to bind into its active site, but the non-competitive inhibitor, right, binds to some site outside the active site, changing the active site's profile so that the substrate can no longer be the substrate is no longer active inside the active site, right? And we're going to go through these in a lot more detail now. So now let's go through each one individually. We have competitive inhibition, right? And in competitive inhibition, like I was saying before, the structure of the inhibitor is very similar to that of the substrate, right? It competes with the substrate for the active site, but the interesting thing about it is that its effects can be reversed by increasing the substrate concentration. So here I have my enzyme, and if normal things are happening, I have my enzyme substrate complex forming after my enzyme goes into the active site, and I get the enzyme end product. And my enzyme is back to normal, back to the way it was before, and it can do more reactions. But if I add the inhibitor, right, I then get the enzyme inhibitor complex. But interestingly, in competitive inhibition, if I add more substrate right here, right, if I add more substrate, you can actually displace the inhibitor. And then I end up with the enzyme substrate complex again, and I'll get the product anyway, right? So that's actually a principle of Le Chatelier's, if you recall Le Chatelier's principle. You increase the substrate so that you get more of the product, so much that it overwhelms the concentration of the inhibitor, right? So here's a more more graphical color picture here. We have the enzyme, right? Notice that the enzyme here is kind of like a trapezoid shaped, right? The substrate is oval shaped, but then it doesn't induce fit, right, model, where now it's all squared off and inside the active site of the enzyme. And then when it's done, everything returns back to normal again, and we get the product, right? But if I add in an inhibitor, this guy right here, it also induces fits with the enzyme, making the enzyme inhibitor complex, right? And then I don't get the product. But if I add more of this stuff, more substrate, then I'll get the product, right? And that's the hallmark of competitive inhibition. Next, we have uncompetitive inhibition. And in non-uncompetitive inhibition, we have the inhibitor binds only to the enzyme substrate complex, right? So here we have EI, all right? And the funny thing is that the funny thing is that when the substrate comes in, right, we still get the ES, the enzyme substrate complex. And the I is still separated, but the I won't bind to the ES, making the SI until the substrate is bound to the enzyme, right? And once that's done, it's useless. Once you have the ESI complex, there's no products being made, and there's no enzyme coming back out again. That is a lost cause now, right? And since that is a lost cause, it cannot be overcome by the addition of more substrate, right? It can't be overcome by the addition of more substrate, right? So in non-competitive inhibition, the substrate can't bind to the active site of the enzyme because the inhibitor, which is a completely different shape, you know, as opposed to what it is in a competitive inhibitor, in a competitive inhibitor, the inhibitor binds to another site on the enzyme that's not the active site, and it completely changes what the active site looks like, right? And when it does that, the substrate can no longer go in there, and you don't get an enzyme back out that's still active or a product. So this is, again, one of those that you cannot reverse the effects by adding more substrate, right? You cannot reverse those effects by adding more substrate. So just a quick review, right? For competitive inhibition, you can reverse the effect of the inhibitor by adding more substrate. Why is that? Because really here, in the competitive inhibitor, the inhibitor is occupying the spot of the substrate in the active site, and if you add more substrate concentration than inhibitor concentration, then it can overcome that concentration of inhibitor. But in both uncompetitive inhibition, you can't overcome it with more substrate, because these are not concentration based, right? Those types of inhibitions are not concentration based. In uncompetitive inhibition, literally once the enzyme substrate complex is bound by the inhibitor, it's no longer useful at all and the enzyme dies, along with any hope of making that product from the substrate. And then in non-competitive inhibition, the inhibitor radically changes the active site of the enzyme. So changing the or increasing the concentration of the substrate won't change that back to normal again. And that's the reason why you can't reverse it with more substrate, right? And then we have one very last type of inhibition, and this is irreversible inhibition, right? And this is the kind of inhibition, again, that can't be reversed, right? And it's when a substrate attaches to the active site, or it doesn't have to be the active site, but attaches to the enzyme permanently with a covalent bond, right? And it can no longer the enzyme can no longer be active because it actively destroys the enzyme, right? And a very good example of this kind of inhibitor is antibiotics, specifically penicillin, which is known as a beta-lactam antibiotic. Beta-lactam antibiotic, right? It's known as that because of this here. We have this four-membered ring here, right? This four-membered ring right there. Let me erase it so you can see it a little bit better. And the interesting thing about it is that there's an amide bond right here in this ring, this beta-lactam ring, right? That's the beta-lactam ring. There's this bond right here, right? That's super unstable, right? This bond is super unstable. Because you recall when you have a carbonyl, right? Its favorite shape is trigonoplanar, right? With a bond angle of 120 degrees. But you can see here that this bond angle is like approximately 90 degrees, right? It's approximately 90 degrees. So it's very small and very strained to what it needs, right? And I'm sorry, this got blocked out right here, but that says beta lactam. See, there's the tam part. So let's add in the lac. Yeah, beta lactam ring, right? And since that's so stable, so unstable, right? It's susceptible for nucleophilic attack, which we haven't talked about before. But basically what happens is that in bacteria, they have this thing called a peptidoglycan cell wall, right? And then there's an enzyme that helps them make this peptidoglycan cell wall, and that enzyme is called glycopeptide transpeptidase. Glycopeptide transpeptidase has a serine right here, right, on its surface. And this serine, remember, has an OH group there, right? And when it gets close to the beta-lactam ring of a beta-lactam antibiotic like penicillin, I'm going to draw this out real quick here. I'm just going to draw the beta-lactam part because that's the business end. All right. Okay. R, right? What can happen is that the electrons right here from this OH group of the serine can come over here and attack this carbonyl carbon, all right? And when that does that, it breaks this bond and it goes over to the nitrogen, right? It breaks that bond and it goes over to the nitrogen. And when it does that, it permanently, covalently attaches itself to the serine. And what it does is that, in effect, it destroys that enzyme. That enzyme is no longer active, right? And that's how beta-lactam antibiotics work. And this is an irreversible inhibition, right? Because the enzyme is no longer inactive. It's no longer active. It's completely been destroyed. And that's how penicillin works. that ends our lecture on enzymes, right? The same stuff is due. At the end of the day today, lecture assignments 13 and 14 and quiz uh, 12. Yes, quiz 12 is due by the end of the day today. I'm always getting confused here because that's a typo. And then at the end of the day next time on April 20th, assignments 15 and 16 and quizzes 13 and 14 are due. All right, thank you, everybody. I hope you enjoyed my lecture on proteins and enzymes. I have office hours tomorrow from 4 to 5 and on Thursday from 4 to 5, so please come by if you have any questions. I'm still working through your tests, so we'll talk about those later. Have a good evening. Bye.